Yeah, go ahead.
Okay, let me share my screen. Okay. Are you able to see my screen?
Yes, absolutely.
Okay. Hello, everyone. Good afternoon. My name is Ananya, and I am a sophomore at Sage Hill School in California. And my research project is about analyzing strategies to enhance the efficacy of immune checkpoint inhibitors for cancer immunotherapy. Okay, so before I delve more into this project, I wanted to address my own motivations for looking at checkpoint inhibitors specifically. While deciding on my research topic, I became really fascinated with immunotherapy once I learned more about what it was. Because instead of a treatment like chemotherapy, where chemicals that kill cancer cells are injected into your body, immunotherapy to me was more fascinating because it involved boosting your own immune system. And while doing further research, I learned that checkpoint inhibitors, a type of cancer immunotherapy, can be remarkably effective, however, only in a very small portion of patients. And I became interested in why inhibitors react differently to different types of cancer and what can be done to overcome this challenge. And so in order to guide my research, I came up with three research questions that I wanted to consider. So I looked at the mechanisms of immune checkpoint inhibitors, the current challenges to checkpoint inhibitor therapy, and some emerging methods to improve their efficacy in cancer treatment. Okay, so the first thing that I researched were the mechanisms of checkpoint inhibitors. T cell or immune cell activation triggers an immune attack. However, T cells also have checkpoints that can deactivate this immune response in cancer cells, and they can use this to deactivate T cells. Now, checkpoint inhibitors are able to block these interactions, including the cytotoxic T lymphocyte-associated protein 4, which can be abbreviated as CTLA4, as well as the programmed death ligand, which is displayed in the diagram on the right. I also learned about a variety of clinical trials that are going on about checkpoint inhibitors. And I learned about how checkpoint inhibitors are less effective in solid cancers as well as cold tumors. So hot and cold tumors refer to the tumor microenvironment. Immune inflamed tumors are also called hot tumors, and they are characterized by very high T cell infiltration. And checkpoint inhibitors are extremely effective in hot tumors. However, immune desert and immune excluded tumors are microenvironments that are referred to as cold tumors. That means there is a lack of T cell infiltration in these types of tumors. And in order for immune checkpoint inhibitors to be effective, the use of other therapies to introduce immune cells into the tumor microenvironment before checkpoint inhibitor therapy is used. And essentially, the goal is to infiltrate T cells into a tumor and turn it from a cold tumor into a hot tumor to make checkpoint inhibitor therapy more effective. Okay, so the process of writing my research paper involved looking into a lot of research papers and ongoing clinical trials. And I learned about biomarkers, which are a way to determine whether patients will benefit from checkpoint inhibitor therapy. Some of these included the tumor mutation burden, which looks at the number of cell mutations, which can also be called tumor purity, as well as the multivariate model, which takes in a variety of factors to determine whether patients will find checkpoint inhibitor therapy to be effective. And I analyzed the need for improvements on biomarkers like the tumor mutation burden to consider multiple factors instead of just one, and the addition of more factors into the multivariate model just to improve the efficacy of these biomarkers so they can better determine how patients will react to inhibitor therapy. I also looked into the potential of AI in the future to analyze huge data sets in order to predict responses to checkpoint inhibitor therapy and eventually even develop treatment plans in a way that human physicians are unable to. I also identified the benefits of combination therapies or checkpoint inhibitor therapies combined with other treatments in order to convert cold tumors to hot tumors. So these are all some of the methods to improve efficacy of checkpoint inhibitors. Now, to conclude, although checkpoint inhibitors have demonstrated success in certain types of cancer, sometimes unfavorable tumor microenvironments have led to limited response rates. And addressing these challenges can lead to the improvement of these tumor biomarkers, such as the tumor mutation burden and the multivariate model, as well as new combination therapies, which is the combination of checkpoint inhibitors with other treatments like chemotherapy, radiation therapy, targeted therapy, or other checkpoint inhibitors. And some of my own contributions involved analyzing the potential of AI to predict results of treatment and develop treatment plans, as well as creating diagrams in BioRender and charts of clinical trials that are ongoing. Now, a quick reflection of my research experience. I had two mentors throughout this process. Professor Virgil was instrumental in guiding us through the process of writing and formatting our research paper, as well as helped us use tools like BioRender, which just enhanced our paper. Dr. Parajuli taught us about the basics of cancer treatment and provided one-on-one mentorship on what things we should include in our papers. And for me, the biggest challenge was definitely reading all of the papers that had lots of cancer-specific jargon and understanding all of that terminology, and then including those papers into my research paper. Of course, I'm not a cancer expert, and so my limited knowledge was a challenge, but it was one that I was able to overcome by dedicating time and working on it. And that's it. Thank you for listening.
Thank you, Ananya, for this great presentation. As you can see, we have two professors. We have dissected writing of the research paper from the learning, which really helps students write the paper. I mean, the biggest outcome that we deliver in this program is a 4,000-word research paper that's worthy of publishing. And that's what colleges look for. You always need to show an outcome. And this program revolves around that. And we have an expert who works just on helping with the paper. And together, they help students not only learn deeply about the topic, but also write a scientific research paper. So great job, Ananya. Thank you for being here.